Hi, I'm Chu, and I'll explain the metabolic pathway in the production of garbage enzyme. The first thing that have to be noted is the production of garbage enzyme is actually based on the fermentation of sugar by the microbes from the fruit peels. The brown sugar that has been added to the sample serves as a carbon source for the yeast, most probably Saccharomyces cerevisiae. After consuming this sugar, the Saccharomyces cerevisiae will undergo alcoholic fermentation under an aerobic condition only. This is the time where the sugar will be converted into ethanol and carbon dioxide. As time goes by, the accumulation of ethanol will favor the growth of acetic acid bacteria. They are responsible for oxidizing ethanol and sugar into acetic acid. However, acetic acid bacteria are obligate aerobes, which means they work oppositely with Saccharomyces cerevisiae. It is found that numerous strains of acetic acid bacteria are capable of survive in the environment where oxygen is not available. It is just that they do not convert the sugars and ethanol into acetic acids. The regular opening of seal to release the carbon dioxide built up in the sample provide a chance for oxygen to diffuse into the sample and enable the conversion of ethanol and sugar into acetic acid. This process is rather slow as the oxygen is a limiting factor here. The strong acid smell that produced by the acetic acid bacteria indicates that the fermentation of fruit is most likely to be completed and garbage enzyme is produced. The enzyme that present in the sample depends on the raw material that we use. This might alter the function of the particular garbage enzyme. The enzyme is actually not produced as a product of fermentation. It is more likely to be associated with the living cells of the fruits. Thus, extending the fermentation duration will not further increase the amount of enzyme in the sample. For example, garbage enzyme that contains papain, which give rise to antimicrobial properties that can be obtained by using papaya as raw material. That being said, enzyme papain will not be available in the sample that use citrus fruit as raw material. Protease, in the other hand, that can be capable of removing protein-based stains is present in the garbage enzyme that use papaya or pineapple as raw materials. Amylase that can be remove starch stains is present in mangoes, bananas, orange peel, and many more. Here, we have an honorable mentions. Although it is not an enzyme, but garbage enzyme that use citrus fruits contains an aromatic compounds called terpenes, which possess grease cutting ability.